It is my great pleasure to introduce Professor Argyris Papantonis, also known to some of you as Akis. So he did his diploma and PhD in the University of Athens in Greece. And then he worked as a postdoctoral fellow with Professor Peter Cook at the University of Oxford. And he was very prolific there. So that in 2013, he was invited to join as a junior group leader in the University of Cologne and was again very successful there. And starting from 2018, he is a professor in the University of Göttingen. Interesting fact about Akis, not many people know that he also writes fiction books in Greek, and actually his first novel, with a very mysterious title, Karyotype, was awarded a literature prize. And he recently wrote a second book as well. Well, his scientific academic writings are also quite known in the community. He didn't receive a Nobel Prize yet, but that's a possibility for future. Well, with this, I would like to let Akis talk about the science. Thank you.
Thank you, Vlad. Thank you so much, and thanks for the invitation. I should probably start by saying that after a year after having published my first novel, I sent it as a gift. So it's written in Greek, right? So I sent it as a gift to my postdoctoral advisor Peter in the UK, and he got it, and then he immediately wrote back saying thank you, and then he said, "How many copies have you sold?" Then I gave him the number, and he says, "Well, have you noticed that this is actually more than the number of citations you currently have?" So that probably says a lot already. So I want to talk today about something that is completely hot off the press. We're hoping to have a preprint in the next week or two or so, but we've been saying this for weeks now. And I'm going to start by saying that my laboratory is specifically interested in the structure to function relationship of human chromosomes. And I've put here a picture that is deliberately blurred to sort of make the point that we understand quite a lot more over the last decade about how chromosomes fold and how this folding affects the different functions that chromosomes have, like replication, transcription, damage, and so on and so forth. But we're still really, really far away from knowing what is going on. So in that effort, we have come, as I said, quite a long way. Now we understand, for example, that there is there's a specific structure in the nucleus of chromosomes. They occupy their own territories, and then if you look within the chromosomes, you can have megabase large domains that are mostly active or mostly inactive, and to some extent they segregate. You can have deep heterochromatin that associates with the nuclear lamina, for example, or the nucleolar periphery, and so on and so forth. So these are hallmarks of organization that we now understand. And people have been going into great detail into what are the forces that actually shape chromatin at really high resolution. So when a particular point, let's say a promoter, talks to an enhancer, and so on and so forth. And to cut a very long story short, what they found is that there are thousands of very prominent loops around the genome that 3C-based technologies, so chromosome conformation capture technologies, can see. And these loops form on the basis of cohesin, and I will come to that a bit later. So a ring-shaped protein, bringing together two bits of DNA that are bound by CTCF and stabilizing very nice loops, perhaps like those shown here. And what happens is, if you remove cohesin from your system, you actually have prevailing active and inactive forces bringing together different smaller compartments of the genome. So essentially, it seems like there is one force which says that homotypic chromatin domains, like active with active and inactive with inactive, like to interact with one another. And there is another force, which is this looping force, that is trying to segregate them. And these opposing forces give rise to the organization of the genome as we understand it today. Now, before I start talking, I want to present the team. So these are three great PhD students in the lab. One more senior, Natasha, that you see here, that has done ATAC-seq plus a number of computational analysis for this project. Shu, who has done all the high C and all the analysis of the high C that you're going to see, and Nadine, who has essentially done everything else. So all the other wet lab work that you will see presented today. And the three of them have been actually working very hard over the last few months to see this project through. So we wanted to ask a very simple question. There is a focus right now in the field on all these CTCF cohesin-based loops that we see genome-wide. And these are very strong and very prominent loops. However, intuitively, you would think that a very important molecule like the RNA polymerase, for example, should be able to bridge active, for example, genes together and make loops, or enhancers and promoters. However, it has been quite challenging to look at these, and people have developed different technologies, which I'm happy to discuss later. But still, there is quite a bit of debate on whether the structural function of the polymerase adds anything to the 3D organization of the genome. And because this has been debated, what we did is we received as a gift a DLD1, so a cancer line from the Mashima lab in Japan, where you can inducibly and reversibly degrade the largest subunit of RNA polymerase II. This is RPB1. And as you can see here, you can just add auxin, in this case for 14 hours. You can deplete RNA polymerase II, but the other polymerases, they're intact, and the cells are still alive. Now, we've done a number of tests to look at what happens when we do this. For example, we've shown that the polymerase, which is also tagged by Clover, really disappears and it doesn't bind chromatin anymore. There's a concomitant loss of H3K27 acetylation, a very well-known activity demarcating mark. And this comes at the expense of more heterochromatic marks apparently spreading throughout the genome. And we've also done nascent RNA sequencing to show the obvious, which is that in the absence of the polymerase, you have a strong reduction in the production of nascent RNAs, and you have lots of genes very, very strongly downregulated. For example, the 560 genes that you see depicted here are actually genes that are suppressed by more than fourfold within these 14 hours. And then what we've done is we've performed Hi-C. So we move away from the 1D component of the genome to the 3D. For those of you that are not familiar with these maps, essentially the diagonal, the two bits, the two triangles that make up this square are identical. The diagonal represents all the positions, for example, along chromosome 21. And everywhere that you see a strong color, so black going to red, going to yellow, there's an interaction. Wherever you see light yellow or white, there's really no interaction. So what we did is we treated these cells with auxin for 14 hours, or added on top of auxin an inhibitor of the RNA polymerase II in an effort to really remove as much of the polymerase as you can from chromatin. And of course, these systems are not 100% perfect, but we can get a very strong depletion of chromatin genome-wide. And as your eyes probably already tell you, there isn't much difference. So very few things happen when you look at these zoom out sort of mega-based resolution maps. So the compartments that I've been talking about, active and inactive, are really not affected in our system if you remove the polymerase. Now, again, you can zoom in into these maps and see finer structure, and you can see very clearly now these dots popping up everywhere. These dots are these structural CTCF-based loops that I was referring to at the very beginning. And I guess your eyes are telling you already that if you remove the polymerase or remove the polymerase degraded and add triptolide, you still have very little effect in the emergence of these loops. And we look for lots of different features, either the emergence of the loops or topological domains, which are sort of submega base domains of interaction genome-wide, or we look at the insulation that occurs between these domains. So this square here doesn't like to interact with that square, so there's insulation in between, and we can look at the score. And honestly, there's very little going on. At the same time that we were doing this work, there was a paper from a group in China, published now in genobiology, where they use mouse ear cells to do a very similar experiment, and they see very much the same results. So all the large hallmarks of genome organization are really not affected when you just remove the polymerase. However, if you really look at high resolution maps and you look at loops, like the ones forming here, we find an interesting subset of loops, about 800, that only appear when we degrade the polymerase, and they are, to our surprise, longer in length than the average length of loops that are there in control, in wild-type cells. You can see an example here. This half is control cells, this half is auxin-treated. You can see, for example, that this loop emerges, that loop emerges, and so on and so forth. And this is the average plot that shows how these loops become really, really strong when you remove the polymerase. And this is a very clear hint that what is going on is that the polymerase was acting as a barrier or counteracting the formation of these loops. And in the absence of the polymerase, the other forces working on the genome allow for these loops to form very strongly. And you can see that at the boundaries of these loops, so the two positions that come together and loop in 3D space, you actually now have a stronger insulation, which means that they come together more often enough and segregate that part of the genome from the neighboring parts. Now, this is all good, but maybe we thought we were looking at the wrong point in time. Now, we know very well that in order for mitosis to occur and the cells to divide, you really need to compact chromatin. So you get this very characteristic shape of the chromosome that we all know of. And if you do a high C at this stage in mitosis, you actually have no structure because everything is condensed. I should say you have structure, but you don't have the structure that is expected of interphase. And then if you just look at what happens in interphase, and this is data not from my lab, from Joop Dekker's lab in this case, from lymphoblasts, you can see very nicely that this very condensed structure that we see here sort of decondenses and gives you all these characteristic domains, either close to diagonal or far away, so smaller and larger domains. We also know that during this transition of the compact mitotic chromosome to the interphase folded one in early G1, transcription seems to play quite a role. We know, for example, and this is work from Gerd Blobel's lab, that there is a wave of hyperactivation of more than half the genes and enhancers in the genome very early as you enter G1. And we don't really know what that is for. We also know from a very detailed description that comes from the same lab of what are the steps of genome reorganization as you go from mitosis into G1. We now understand that there are many events that happen early before we start seeing this characteristic cohesion CTCF loops that I showed you before. So could it be that the RNA polymerase is important there? To test this, we devised a very simple scheme. We took cells and synchronized them at the G2M checkpoint that you can see here using a Rho3363 inhibitor. At that point, we added auxin to degrade all the polymerase from these cells because the polymerase is not part of mitotic chromatin, and we then released them through mitosis, allowed them to quantitatively re-enter G1, as you see here. So the vast majority of cells, 6 hours after washout, are actually G1 cells. Sort out, using FACS, these G1 cells and perform high C on them on cells that have or don't have the polymerase. So essentially, we allow cells to enter G1 by having or not having the polymerase. We can do fractionation of chromatin and check to see whether the levels of different chromatin-bound factors changes. For example, you see the clear reduction of chromatin of the large subunit of the polymerase, of activated initiating polymerases marked by serine 5. On the other hand, we don't see much of a change for CTCF, but we do see changes, for example, in these chromatin remodeling factors. So everything worked the way we assumed it would. We also see, and I'm not going to go much into detail here, that we have a loss of local interactions happening within one chromosome in favor of really long-range interactions between different chromosomes. And some of these we verified with the help of the Rukos lab in Mainz using 3D DNA fish. And this is where the surprises started happening. So this is the control map at very low resolution, and this is the auxin-treated cells. And I think you can already see that the definition of the compartments is very, very blurry, and there's lots of mixing between compartments that otherwise should be very well defined and segregated. This means that active and inactive compartments start to interact with one another. That wasn't the case before. If we zoom in at the level of TAD, so at the level of sub-mega base domains, I think you can clearly see that we have an erosion in the structure of these domains. Everything becomes much weaker. The definition at the boundaries within this large domain starts to disappear, and also these dots that we identified before as loops go away. This is a very strong effect. For those of you that are not familiar with high C changes, this is one of the strongest changes you can see in one of these degradation-like scenario. Here's another example of this erosion of domains and loops that we see. We also see some new loops, some new domains arising, and these are larger in size. And we attribute this to the loss of boundaries between domains that allow these domains to merge and appear in our analysis larger. But this is not the big thing. The big thing is that if you go and look at high resolution, where we expect the polymerases to act and form loops, we actually see that more than 2,000 loops in the control cells completely disappear, cannot form without the polymerase, and about 1,200 new loops that are also larger, just like before, appear de novo. Here is an analysis of the shared loops, which, although are still detectable in the absence of the polymerase, they are far weaker. And you can see, you can see that the insulation at the anchors of these loops also becomes weaker, which means that in a significant probably fraction of cells, these loops either form very weakly or do not form at all. So overall, there is a weakening of looping in this scenario, and also new elongated loops start popping up. We also did an experiment that, again, mostly serves as control, and I'm going to very quickly go through it. And the idea here is that we did the same in a line, another cancer line, HCT cells, that have a full knockout of topoisomerase 2B and a degradable major topoisomerase 2A. And this was to show whether you need the topoisomerase, so you need the actual activity of topoisomerases to form these loops. And if you were to, for example, remove this activity, would you get the same results? And we went through the same FACS sorting and cell arresting protocol like before. We checked for genes going up and down. There's very little effect, surprisingly maybe, on gene expression. But looking at any feature that you might imagine on these high C maps here, high and low resolution, we really don't see differences. And even the small differences that seem to arise at the level of loops, as you can see here in average plot, they're very, very marginal. So essentially, we can deduce that it is not a supercoiling effect that we're mapping here, at least not to a great extent, and whatever changes we're mapping are mostly centered around the RNA polymerase. We went back to our fractionation protocol and looked for reduction of cohesin-associated components. You can see here, for example, that the two of the main components of the cohesin ring, SMC1 and RAD21, seem to be equally embedded in chromatin, regardless of whether you have or you don't have the polymerase. But at the same time, NIPBL and MAL2, which are the major loaders of cohesin onto chromatin, they are significantly weakened in the absence of the polymerase, and so is WAPO. We verified, for example, using fluorescence, is just an example, that indeed RAD21, so cohesin, seems to be on chromatin to the same extent that it is in the control cells as it is in our auxin-degran cells. And then, in order to see whether this had anything to do with genome organization, we turned to super-resolution localization using DStorm. This was done in collaboration with Johan Slotman and Kirsten Wendt at the Erasmus in Rotterdam. And we've done two-color storm looking for NIPBL, which is the loader of cohesin, and CTCF, which are the molecules at the base of which cohesin stops to form loops. This is what they look in the control cells. You can see nice distributions, nice round clusters forming in this very thin section of the cell that we're imaging. And then if you look at the oxygen-treated cells, there are a number of things to unpack here. First of all, you start seeing these elongated, peripherally positioned NPL clusters that look very non-canonical. And at the same time, you see this dispersion of CTCF mostly in smaller clusters. I should say that the only other degradation experiment that gives you this sort of a result is when you really deplete cohesin itself. So when you degrade cohesin itself, you have a loss of loops, dramatic, genome-wide, and you have a very similar storm imaging result like this. Again, we have a change in the mean size of the NPL clusters, although we have many larger ones. The majority of them actually decrease. We have many more CTCFs now forming smaller clusters, which could be indicative of them disengaging from their looping position. And we also have a vast change in the separation, relative separation between any two closest NPL and CTCF clusters. So this implies, without us being able to pinpoint where this happens on the genome, obviously, this implies a strong reorganization of NPL and CTCF and perhaps CTCF-based loops genome-wide. At the same time, if you just chip SMC1A, which is a cohesin subunit, as I said, and look at whether it ends up at CTCF sites as it should in order to form loops, you see that this is greatly reduced. And at the same time, by analyzing these two data sets in control and oxygen-treated cells, we find that there's lots of added noise to this data set, as if there are many cohesin molecules now that are floating about on chromatin without ending up at CTCF sites. These are not easy things to interpret and put together. And in order for us to do so, we turn to computational modeling. And this was done in collaboration with Davide Maranduzzo in Edinburgh, who's a long-term collaborator of ours. And what he did is he first used his hip-hop model, which is a very cool way of modeling data. This is a 2018 Molecular Cell paper, if anyone wants to look at, and essentially found that if he disallows loading of cohesin at promoters, like our data probably imply, you have a collapse in the structure, also shown by the models here, that is very similar to the one we're seeing in our high-C data. However, the really interesting results come when you look at loops in this region. We see a very strong weakening of loops, exactly like we see in the high-C data. We see fewer loop events. You can see the frequency here going down. And perhaps to our surprise, we see an increase in loop length in this 15 megabase region that we're modeling. I should say here, for full disclosure, that when these simulations were done, we had not looked at the length of the loops because we could not imagine that the new loops that form are actually larger in size. And after the simulations gave this result, we wanted to prove or disprove them, and we looked into the data. And again, to our surprise, found that all the de novo loops that form in the absence of the polymerase are indeed larger. Now, you can do another type of modeling, which is much simpler, but also very cool. You can actually take a 3 megabase fiber and only model two forces on it. So one force is the RNA polymerase transcribing genes, and you can see here the genes exactly as they are on the fiber, here on the left, the control scenario. And the other is extrusion of loops by cohesin. So essentially, the ring of cohesin pulling chromatin to make loops. And these forces, in some places on the fiber, they work together. In other places, they work against one another. And you can see that in an experiment that you can only do in silico. We turned all the promoters facing the same direction, and you can see how they reshape the partitioning of this part of chromatin here. So being able to see that we can model these competing and converging forces, we moved on to simulated degron, which is not allowing cohesin to load at the active promoters, but only randomly across the fiber. And you can see how you really lose structure, much like in a high-C data. And again, the frequency of looping really goes down, and whatever de novo loops form, they're actually, on average, larger in size. And this is the last slide of data. as I said at the beginning, you have, when you remove the polymerase from an asynchronous population of cells, you lose K27 acetylation, you don't have the polymerase there, you don't have transcription, and as expected, you can see here in 2 replicas, you have less accessibility. So it could be that a loss of accessibility also in our G1 reentry cells would actually mean that it could be just accessibility. Cohesin likes to load at positions that are just accessible, and you don't have the polymerase, accessibility goes down, and then you can't load cohesin, you don't get loops. However, our ATAC-seq data beg to disagree with that. These are the control reentry cells where you can see accessibility that is, let's say, comparable or similar to what we have here in the control cells. This is accessibility that is made de novo as the cells enter the cell cycle. And then when you use auxin and the cells enter the cell cycle without the presence of the polymerase, it seems that there is aberrant opening up of chromatin at these same positions. This is looking around TSSs where we suppose that cohesin is loaded. And actually you have, if anything, equal or more accessibility at these sites. So it cannot just be the accessibility that allows the loading of cohesin and the formation of loops genome-wide. It must be something that has to do with a component or cofactor of the RNA polymerase II. This then means that the idea of cohesin being loaded onto DNA, like you see here in the model, using RNAPII, and then start to extrude loops at a particularly high speed, and then stopping when it finds these 2 CTCF sites, needs to be revisited. And we revisit it as such. We assume that the loading is not random, and especially as cells re-enter, after mitosis, re-enter the G1 phase of the cell cycle, they choose to be recruited and loaded at sites where you have active RNA polymerase. And these could be promoters, and they could also be very active enhancers. And the recruitment requires RNAPII. In the absence of the polymerase, you do not have efficient loading, and a vast portion of the 3-dimensional organization of the genome collapses. So this was it from my side. I'm a minute over. I would like to thank the people that are paying the bills over the last few years. Davide, Vasilis, and Kirsten, without whom we couldn't do any of the modeling, the 3D fish, or the super resolution localization. And again, Natasha, Nadine, and Shu for all the work they've put in over the last months. And I'm happy to take questions.
Thank you very much for this excellent talk. It's a great talk. Now let's see whether we have questions, whether we have people who want to ask them by voice. So we have Job Decker raising hand, so I unmute. Can you unmute yourself and ask?
I should be unmuted now. Is that right?
Yes. Thank you.
Okay. Thank you. Thank you. That was a fascinating talk. Lots to think about. I was wondering how you selected your G1 cells. Because there's a lot of changes in folding between early and later G1. So how do you ensure that the with or without auxin case are in the same point in time in G1?
This is difficult to say. We do not really know if they're at the exact same point. The way we've done it is probably slightly naive. So what we've done is we've used the control cells, and we've done a time course that goes allows the cells to go essentially past G1 and into start going into S phase. And we time that very carefully. Nadine did this, and then we repeat the same time course with the Degram cells, and we get a very similar profile. We collect the cells when they start accumulating in G1, so there are far less, I don't want to say no chances, but there's far less chances that they all just represent very, very early G1, where indeed you have we start build up the structure and some of the features are not there. And if you look at our maps and compare it, for example, to the really high resolution stuff that you had in the Nature Cell Biology paper, or Jennifer Kremens and the Global Lab have in their Nature paper, they don't look like very early G1. Not at all, actually. This is the thing that put us at ease.
Yeah, the compartments are very weak early G1. But another question
they are, yes,
related. Yes. You use the R0331, I believe.
R0336, I think, yeah.
Polykinase, so you're in early prophase. So you also have no polymerase during mitosis. Do you think it would difference if you would degrade Pol2 in metaphase, like in mucosal or best?
Yeah, it is. So that Nadine tried the Nucleosome. It doesn't work very well with the DLD1 cells in our hands. That was the reason why we didn't go that way. It would be interesting to do it later. It is very tough to remove lots of the polymerase from chromatin. So we now can remove with this scheme around 90%. We had one experiment, and I should say this is also a sort of funding thing because Hi-C, as you very well know, is expensive to sequence. We had one experiment where the degradation didn't work as well using the same scheme, and we don't see such strong events. So we had, I don't know, about 70% degradation, and the effects were not as strong. So you really need to remove as much of the polymerase as possible. And this also is a bit telling about using inhibitors that are never able to remove more than 70–75% of polymerase from chromatin.
Yeah, thank you. That was a fantastic talk.
Thank you, Jan.
Thank you. Great. Our next question is also by voice. So there is a hand by Mark Martirino. So Mark, can you unmute yourself?
Thank you, Vlad. Do you guys hear me, I guess?
Yes, yes.
Okay. Hi, Akis. Thank you very much for your talk. I wonder, I mean, many of the analyses you have performed from your data requires that you select loops, that you detect loops from the Hi-Cs. And I wonder what could be the effect of using different types of software. You know, we developed this type of software, so I'm very much interested in you have checked whether using different types of software will give you slightly different results or not, or whether your results are very much reproducible if you, let's say, change the software for detecting loops.
So this has been a concern, and we went through a number of them. We started by using, for example, tools embedded in Juicer, which is what we started using in the beginning. The detection there wasn't as robust. We've tried a number of different things at different cutoff values and resolutions. The one that worked best in our hands, and by best I mean that it was robust and not also affected a lot by the sequencing depth, which can make a huge difference. If you have 400 million reads or 500 million reads, some piece of software are really affected. So it's the one called SIP, SIP, which seems to, so it essentially uses the image of the map to look for the loops, and it seems to work very well and pretty robust, even if we switch the resolution from 10 kbp matrices down to 5 kbp matrices. So that has been good. We want to try chess now, well, you know what chess is, that just officially came out. And we are hoping to, so we've already tried 3, I think, methods to do this, and SIP was the best. There is a dependency on the method. There are some things that we keep seeing. And I should say that the collapse that we see in the reentry cells in the absence of the polymerase is actually so strong in most of the places in the genome that you don't have any of these software failing to see it. But you might have 100 loops more or less called in the different conditions if you just look at asynchronous cells, for example. So this is true, and we sort of compromise on that a bit.
No, thank you, Akis. Very reassuring if you see this is strong. Thank you.
Thank you very much. Our next question, I'll read it from the Q&A box. So Konstantin Milonas is asking, have you tried a high chip with antibody against RNA polymerase to ensure that the loss of loops after auxin treatment is due to a direct depletion of RNA Pol2 and not because of secondary effects?
The short answer is no. The longer answer is that in the genome biology paper that I mentioned, where they use the mouse cells for simple asynchronous populations of mouse cells, they did K27 acetylation high chip, and that worked very well in their hands. And they can see that there are many loops that are enhancer promoter loops, for example, that collapse. People in the past have used inhibitors and, let's say, capture-based technology and have seen some change. We have focused mostly on the idea of re-entry into G1, and there we were less interested in looking at high-resolution loops because between enhancer promoters that would require high chip. I should say that in our hands, and I say this with a bit of caution, but in our hands, we have had trouble getting very, very complex high chip libraries, and we've tried for CTCF and K27 acetyl. So given the amount of high C that we've done so far, I've been reluctant to try this. Micro-C could also be a very nice alternative here, but we don't have it set up in the lab.
Great, thank you. Next question from Peter Sarkis. He's asking, could it be that the presence of Pol2 on chromatin that is important rather than ongoing transcription?
I think that is exactly the case. I think that the elongation is not the case. I think the topoisomerase experiment speaks for this, and that it's the presence of chromatin at promoters and enhancers, I guess, that somehow recruits cohesin, allows to with particular rates, and allows it to translocate and extrude loops. This is what we think. This is the current scenario.
Thank you. Question from Bharath Saravanan. Is there an enrichment for Pol2 at the loop anchors that are gained?
The ones that are gained, so this is a difficult one. So some of the loops that are gained in the absence of the RNA polymerase are actually loops that form due to polycomb. And there is work from Rob Close and Juan Mavaquerizas that show that loop extrusion, so cohesin, essentially is counteracting interactions between polycomb domains. And when you don't have it there anymore, you can see such looping. And we actually, for a small subset of the loops that we're looking at, for example, around the Hox loci, which are famously covered with K27 trimethylation, we do see such loops arising. For the rest, because we see about 1,000 such de novo loops forming, we are looking into the strength and orientation of transcription as we speak, so I don't have a good answer. There seems to be a bias, but I'm not certain enough to comment on it yet.
Okay, great. Thank you very much. So we have to actually, okay, let's ask another question. Okay, let's ask one more question. A question from Nicholas Sapp, who says, great talk. Does your work suggest a direct interaction between RNA Pol2 and the cohesin components?
It does, and it does also suggest that the loading, at least while re-entering after mitosis into G1, may rely on RNA polymerase II on chromatin. We are trying to test this further. Again, it's not easy, right? I mean, the polymerase is doing many things on chromatin that one needs to be careful, but it seems to be the case.
All right, great. Thank you very much.